Okay. Yeah, it'll be me with a quick introduction of the Work Package 5, which is actually not really the equivalent of what Andrea has shown to us, was much reduced effort. And then will be Robert showing very quickly the major QTL we found in sea bass against this one. Yeah, that's easy. And then to conclude, Sarah, she will show the very first results of using genomic information, functional genomic information into using for genomic prediction. So this very simplified scheme that illustrates one of the major goals in applied genetics and genomics. You have millions of variants, genetic variants. Most of them are bystanders. They don't have any role in the phenotype of interest. And there are very few, sometimes thousands, but still a very small fraction compared to the millions of the remaining, which are actually causing or contributing to determine the phenotype. And so clearly, identify those causative variants is the holy grail of genetics in a way. And as you can see, this is the point. Yeah. So they could modify the protein coding sequence. So you might have a major or a minor effect on the protein function. And those variants are relatively easy to predict in silico because we know, or now we know even better, the protein coding genes in our genomes. But and you can predict the effect of individual variants. But the big black matter is here because the regulatory elements are not easily just to find, not to say to predict the effect of the variants within those elements. And those are actually probably less important effects, very light, but probably the most important. There is an estimate, maybe it's an overestimate, in human genomics that major complex phenotypes, actually the underlying genetic variants are regulating gene expression rather than changing protein coding genes. So unfortunately, the most of the time and the resources had to go to characterizing those regulatory elements in Aqua Faang. That's the limitation of Work Package 5, time and resources. In fact, as Dan and then Peter has shown us, we identified a massive catalog, which is now publicly available, which is, I think, a great result. So what we tried to do in Work Package 5 is to link genotypes, phenotypes in fish, and functional genomic information, which, I think this has been already mentioned. There are three major potential applications. One is fine mapping relevant QTLs. The second one, Andrea was explained very well to us, improving overall, including all the information, improving the overall genomic prediction accuracy. And finally, it was not touched actually in Work Package 5. The third and potential application is having knowledge-based genome editing. So what we did actually is to work with what we had already. So for some species, we had a phenotype and genotype collected in previous project. And so we overlap functional genomic information collected in Aqua Faang and to refine, in a way, QTLs, like, for instance, QTLs for disease resistance in sea bream, same for turbot, and so also for common carp. We only had actually one, and these actually there would be, they will turn into improving the SNP arrays, SNP panels that will be presented by Selective just after this talk. So we had only one major case study, which was VNN and resistance in sea bass, where we started from the experimental challenge. We had eQTL information, we had whole genome sequencing, and we are we will see we find a major QTL. We are still at the very, very beginning in testing the genomic prediction accuracy increase. So if Robert can come.
Thank you, Luca. To test the genetic background or the genetic architecture of VNN resistance in sea bass, so we had this large kind of disease challenge where we had 25 males and 25 females crossed to produce a large population of about 1,000 individuals, which were challenged by the NNV or the nervous necrosis virus, and then another proportion of about 300 individuals which were used for the functional, for generating functional information, including RNA-seq, attack-seq, and also some small RNA. So all this information will go back into genomic prediction, but just a little bit about the disease challenge. So all the individuals were individuals in the genomic kind of challenge or the genomic study. Individuals were challenged for about 26 days, and the phenotype we recorded was binary survival. So if the individual dies during the 26 days of the experiment or survives at the end of the experiment, or days to death, time to death, meaning the time it took for the individual to die from the time when we exposed them to the virus. So we genotyped all the individuals, parents, and the offsprings from the two challenges, and on a 29K SNP panel that is currently available, and then we full sequenced the genomes of the parents and I think 40 of the offsprings to generate a reference population that we used to impute the entire population that we used in the study to full genome genotypes of about 8 million genotypes that we used in EQTL analysis and also in GWAS, and we will proceed to use them in genomic prediction. So a little bit of the results. So from the challenge study, we saw that it worked quite well, and around the 4th or the 6th day of the challenge, we had a maximum mortality, and then it dropped. And the challenge study was stopped when we saw no more deaths. And from the cross, we see that indeed all the dams and the sires indeed contributed quite well to the population that we studied. So when we reconstructed the pedigree and we looked at the phenotypic differences in terms of resistance to the virus, we see quite good variability in terms of resistance phenotypically within the families, within both the sire and the dam families. And that kind of variability, since you see it within families, it is an indication of genetic variation in terms of resistance to the virus. So indeed, from the previous slide, it is not surprising that, we see quite substantial level of additive genetic variance, which is expressed as heritability. So we see quite a high level of heritability, meaning to the breeders that this is something we can breed for and indeed sustainably reduce the impact of the disease to the sea bass aquaculture industry. So going to the GWAS results, very impressive results. I think we are all proud of the result. they are impressive and convincing. So we are pretty sure that we got it. So looking at, we got that major QTL. Indeed, when we looked at the variance explained by the variants that we identified as the most significant variants, indeed, they explain up to almost 40% of genetic variance. And when you look at the population clustering in terms of resistance, you see that there is a significant difference between the individuals that have the resistant genotype of the QTL compared to those that have the susceptible genotype of the QTL. What I've just said, 90% of the individuals that had the resistant genotype survived. But the problem we see that indeed the population we used, the individuals that have that impressive or quite what we would want in the population are quite low, and that means there is room for improvement. So that QTL, which indeed, so on top of the GWAS, we also did some quite functional analysis in addition to this. For example, EQTL analysis and also a toxic analysis. And we see that the most significant markers are located within a region that is harboring a lot of genes that are interferon-inducible genes that are quite well established to be involved in immune response, especially to viral infection. So to further pinpoint what are the probable causal effects in terms of in silico analysis, so we have quite refined the QTL and the genes that we believe are the causal genes. So it is a cluster of these interferon-inducible genes, 3 of them, which we saw that they had quite a high correlation with the phenotype or the breeding value of resistance to VNN, and also with gene expression. So their gene expression levels are quite close to the phenotype in terms of correlation. So with the ATAC-seq results, so we saw that two of the most important or the most significant SNP markers are located within the transcription binding site of these 2 genes, and they according to an in silico prediction, indeed we saw that these 2 positions or these 2 variants have got an effect on the binding sites of these 2 genes. And we are somehow suspecting that this could be the mode of regulation of these markers to the gene expression, and probably also correlating with the phenotype, because the SNPs that we identified in terms of regulating gene expression in the EQTL overlap exactly with those that are having the most significant effect on the phenotype. I think that's me. So, thank you.
Yes. Is it? Yeah. Can you hear me? Yes. Okay. So yes, we conclude this part of the work package 5 of the Aqua Faang, trying to place all the information that we got from Andrea at the beginning, and then from Luca and Robert, into the aquaculture context. So yes. Yeah. So just to provide a little bit of background, I know that most of you already know, but why we are interested in selective breeding. It's because we can provide a cumulative and permanent improvement in the trait of interest. So all the traits that we are interested in. So, yeah. And why selective breeding could be really powerful for aquaculture species is because aquaculture species have some features that are very peculiar to those species. So we have high fecundity of fish species with a large stock productions. We have small generation interval, and what is really important for breeding is that we have phenotypic and genetic variability in the trait of interest. So these are all peculiarities of the aquaculture species that give us very, I mean, it's very good to, it's a very good results for selective breeding. So so selective breeding and all the relative improvements in the traits could lead to a progress and sustainability of intensive aquaculture systems. So it's really important to going forward. The breeding goals in aquaculture are very similar to those of terrestrial species. So we are interested in improving disease resistance, in improving productivity, improving efficiency and quality traits. But what is really important for these traits is that routine individual phenotyping for these complex traits is really difficult. And normally, in traditional selective methods, so when we estimated breeding values of the animals, using traditional methods, we need routine individual phenotyping. So we need to collect phenotypes every generation or every 2 generations. So it's really difficult, and we need, for these traits in particular, to other methods, alternatives to these methods. So, okay, yeah. So these alternatives are available, actually, because we need SNP arrays, so we need genomic information, and we have, for aquaculture species, we have SNP arrays covering from thousands to millions SNPs. We can individual genotype with feasible and reasonable costs. And we have also whole genome sequencing. So we arrived also at the information at whole genome. Oh. So using this genotype information, genotypic information, we can use marker-assisted selection. So we have already seen that. We can use the information of major QTLs associated with some traits. This is just an example in rainbow trout. We have already seen for European sea bass that we have also a major QTL. So we can use this information of major QTLs, so quantitative trait loci with large effect in marker-assisted selection. But what I want to stress is that it's the exception, it's not the rule. So we can detect some major QTL for the trait of interest, but it's not. It's not. I mean, it's not always like this. So commercially important traits are normally characterized by many QTLs with very small effects. And in marker-assisted selection, few genetic markers are exploited, which are those of the major QTL. So we have low performances for polygenic traits characterized by many QTL of small effect. So what we can do? We can go to genomic selection. So we can estimate the SNP effect and we can select the animals for breeding based on their genetic merit. And this genetic merit is predicted from the joint effect. So we can combine SNP effects, and we can predict the genetic merit of the breeding candidates, and we can select the breeding candidates in this way. This is just a very brief example of how genomic selection works, genomic prediction works. What we have already seen from Andrea, that's how it works. So it's just a very brief reminder. So we can use phenotypes and genotypes of a reference population. We put everything in a model, and we can use this information to estimate SNP effects. And then we can use the prediction equation to estimate genomic breeding values of the breeding candidates. So genomic selection has very good advantages, has main advantages. So it's suitable for traits that cannot be measured on breeding candidates. So all the traits that require a challenge or require the slaughter of the animals can be improved using genomic selection. Phenotyping could be performed only once on reference population. We will see if this is really true or not. We can assess the individual genetic merit for the trait of interest. In traditional selection methods, normally we can assess a family genetic merit. But with genomic selection, we can do it individually, and we can anticipate the selection of breeding candidates. So this is really important for farmers because we can really anticipate the selection of the breeding candidates. So I was saying, is it true that you can phenotype only once? What happens in reality? What we have seen is that there is a reduction in genomic prediction accuracy when the relatedness between training population, so the reference population, and animals to be predicted are not so related. So these are just some examples in the literature. So we saw that for sea lice resistance in Atlantic salmon, for VNN resistance in sea bass and in gilded sea bream, the relatedness between training population, when the relatedness between training population and animals to be predicted is low, we have also low prediction accuracy. So trying to place this into the application perspective, we need prediction equation that can perform properly even when the reference population and the candidate breeders are genetically distant. This means across generation or across populations. So we really need to phenotype only once and build a prediction equation that can perform properly across generations. So how we can improve genomic selection with this in mind? And this is what we have already seen at the very beginning of the day from Dan. So we can use the genotype efficiently to predict the phenotype or the genetic merit of the breeding candidates. So we can take advantages of causative variants, even those with small effects. And we can try to understand if these causative variants are located or not in regulatory sequences, because we know that they can affect the trait through changes in gene expression. So we can use all this information to filter or to integrate this information into prediction models. So we can filter the genetic markers according to their localization inside functional sequences. So this is just a very brief presentation of the Faang project. So from Faang projects in general, but from Aqua Faang also, we obtained a lot of information, a lot of data sets, so functional genomic information, expressed and regulatory genomic regions, and annotated genomes for farm animals. So also for farm animals, not just for model species, but also for farm animals. How we can use that? We can relate genotype, phenotype, and genetic merit. We can predict genotype-phenotype relationships, and we can also use genotype efficiently to predict the phenotype. And this is really related to the application. So we can really turn the research outputs into industry application. We can improve and facilitate genomic selection, and we can go forward precision breeding. So this is just the experimental design we have already seen from Robert. It's absolutely the same. So our target species was European sea bass. The trait of interest is resistance to viral nervous necrosis. And in this case, we try to understand if we can integrate genomic functional notation to improve genomic prediction accuracy. The experimental design is the same. So we had this experimental population. It was challenged. The trait is a mortality. We genotyped all the animals, and also we imputed to whole genome sequence all the offspring, so all the experimental population. And we also tried to detect active regulatory functional elements in the genomes, so the so-called open chromatin regions. Just to put some other pieces in the puzzle, we tried to prioritize SNPs using functional information. We predict the breeding values of the animals for BNN resistance. And we try to validate our prediction equation using training and testing sets consisting of animals as genetically unrelated as possible. So we will see later that is quite, well, they are not so related, but I mean, the families are produced using the same parents, so we'll see. So this is just the ATAC-SEC analysis that we performed, so we have already seen that. In our case, we had two conditions, so the mock-infected animals and the nodovirus-infected animals. And for each condition, we sampled two tissues, so the brain and the head kidney. The brain, because it is the target tissue of the nodovirus, of the virus, and the head kidney, because it has a very important immunity function. And this information were used to perform ATAC-SEC analysis. So the output from the ATAC-SEC were used to pre-filter the whole genome sequence. So we tried to detect SNPs included in the open chromatin regions, and we retained just these SNPs. We created different subsets to be used in genomic prediction using the different combination of condition and tissues. We have already seen that a major QTL has been detected, so this chromosome has been removed, because in this case, we wanted just to see if functional annotation data could be useful in genomic prediction. So I was saying we detected two different groups. Okay, two different groups, but they are not very unrelated. I mean, they are produced using the same parents. But we tried to use these two groups in a twofold cross-validation, so trying to minimize the relatedness between training. Sorry, it's the opposite. So maximize the relatedness between training and testing, and minimizing the relatedness within sets in a single-step genomic lab. So these are just some preliminary results. These are on the left. You can see the correlation between estimated breeding values, so using the pedigree information, and the genomic estimated breeding value obtained using the methods I explained. So you can see that more or less the correlation, these are just person correlations, so it's more or less 0.37. On the right, you can see the accuracies, and they are more or less 0.34. These are the correlation between the GBV obtained using the partial data set, so using the two-fold cross-validation, and the GBV obtained using the whole data set, so without masking any phenotype. And you can see that the correlation are more or less 0.39. So just to try to understand and to see what are the future perspective, Aqua Faang has allowed the generation of a huge amount of data. So these were just very preliminary results integrating ADASEC output into genomic prediction. But we have also ChIP-seq data, EQTLs, chromatin status scores, and some regulatory data. So these could be other useful criteria that can be used to give a weight or to filter the genetic markers. Anyway, the integration of this functional information data could be useful to produce new genetic tools and selective breeding approaches that could have better accuracies. And this is a step towards precision breeding. So it's something that it could be very, very useful for farmers. Accurate prediction of the genetic merit of breeding candidates can be obtained even if the reference population is distantly related to the breeding candidates, so across generation or across population. This is what we aim for. So we will see if using other data we could do that. And another important point is that we need to reduce the need to collect phenotypes on complex traits because it is really costly, it is time consuming. So for these complex traits, we need to reduce the need to collect phenotypes every generation or every few generations. In the end, the point is we need to optimize the resources. So we have to improve profitability and sustainability of the aquaculture sector, and this is the final aim of the Aqua Faang project. So we will see if we will be able to do that using all the data sets that have been generated in Aqua Faang. So this is all by me, and I would like to thank all the people of the Aqua Faang consortium and you for the attention.